Hi students, I am Shivangi Tripathi. So today I'll be teaching you monohybrid cross from the chapter genetics of class 12. This means that one character is considered for the hybridization of plants. For example, the height of the plant, tall or dwarf, and second is the flower color. It can be violet or white or red or white. Okay. So in this example, I'll be considering the height of the plant, that is tall and dwarf. So let's see. See in this the alleles that are to be considered, that is of tall. For tall character we considered capital T, and for dwarf character we take small t. Okay. Since this is a parental generation, so we show it in this way. Capital T, this is a homozygous dominant phase of tall plant, and this is homozygous recessive phase of dwarf plant. Okay. Then while crossing over, we will first see what are the genes are to be made in this from this parental generation. The genes would be capital T and small t. Okay. So in this, from by using these alleles, these are alleles or genes. Okay. By using these alleles, we will create a Punnett square. In Punnett square, on this side you can take the male alleles, and on this side you can take the female alleles. Okay. So by crossing over, by cross multiplying, you can do this type of cross. Capital T into small t, this will make capital T and small t. This thing, it codes for the tall plant. Why? Because in this the capital T is present, and capital T is, that is for dominance. Okay. The character which is dominant in a gene, that only character is expressed in a phenotype of the plant or animal. Okay. And the other one, small t, this shows the recessive character. Recessive means this character is hidden, or you can say recessed, or this one is dominated by the dominant allele, that is capital T. Okay. So this Punnett square, in these all the plants, they are tall. See, this is called as heterozygous dominant tall plants. Okay. So in F1 generation, if we will see, so we can see all the plants are tall with the genotype capital T and small t. Now students, coming to F2 generation. For F2 generation, what we will do is we will selfing is done of the tall plants of the F1 generation to see whether this genotype is heterozygous or homozygous. Heterozygous means this one, capital T and small t. Homozygous means this one, capital T and capital T, small t and small t. So this one is heterozygous and this is homozygous. Okay. So in F1 generation, this genotype, now this genotype we will consider it as parental plants. Okay. With heterozygous genotype, capital T, small t, crossed with capital T and small t. Selfing means the two plants from this generation, from F1 generation, we will cross, means then this is also called as selfing. This downward arrow is, this thing stands for selfing. Okay. Selfing means self-pollination. Okay. For F2 generation, we will draw we will draw a Punnett square. Now in this case, see here I took all the alleles of a male plant as in this direction, and female in downward direction. But the case here is different. Why? Because here this is homozygous dominant alleles, but this one is heterozygous dominant. So these alleles we will place it over here in two different columns, capital T and small t. Similarly, in this column, okay, capital T and small t. Now we will cross multiply these alleles. Capital T into capital T. This codes for tall. This one is homozygous dominant tall. Next one will be capital T small t. This one you have to just cross multiply simply like this. So this is capital T and small t. This codes for heterozygous dominant tall. Now again this will be same capital T and small t. This one will come again it will come as tall. This one is heterozygous dominant and this one is homozygous dominant. Now this one small t and small t dwarf. Students, this is the case in F2 generation. This shows that the parental generation and F2 generation, they were heterozygous dominant. Why? Because in F1 generation no plant with the small phenotype was shown. But in F2 generation, after selfing or self-pollination, we found out that 25% of the plants, they were found to be dwarf in the phenotype. Phenotype means the physical characters. They were short in their height. So this we call it as homozygous recessive. Okay. So based on this, we will now find out the ratio of the genotypic and phenotypic characters. Genotypic ratio means that the ratio of these genes, the alleles that we have crossed over here, capital T and small t. Okay. So what we will do is we will place all the different types of hybrid crosses that we have made. This is homozygous, heterozygous, and homozygous recessive in one line. Okay, and then we will count the number of plants formed related to this genotype, related to homozygous dominant, heterozygous dominant, and homozygous recessive. Then we will just place the number of plants beneath their genotype. So this will be your phenotypic your genotypic ratio. See, since in F2 generation, this one is only 1 in number. This is dominant homozygous. So this is only 1 in number. So we will write 1. Now heterozygous dominant, these are 2 in number. So we will write 2. And this one is 1. So 1 is to 2 is to 1. This is the genotypic ratio of F2 generation. Now coming to phenotypic ratio. Phenotypic ratio means the physical characters of the plants that are being shown to you. Okay. So we can see that 3 out of 4, 3 plants, they can be seen as tall. 1, 2 and 3. And 1 is dwarf. So we will write tall is to dwarf, 3 is to 1. Why I have taken tall to at first place? Because the dominant character is considered always at the first place. Then the next recessive character is considered. In this case, genotypic case, both these are dominant. But homozygous dominant is taken to be is given priority, and then heterozygous dominant, and then in the last homozygous recessive. So phenotypic ratio came out to be 3 is to 1. Okay. Well, thank you students. That's all for today.